Now, okay, so we're recording. So welcome, Brit. I will pass over to Mick to do the introductions. Just quickly, if anybody else is watching just during the seminar itself, if you could mute your microphones. I can, I think, do it from my end, but it's probably better if you could do that yourself. Yeah, so take it away.
Okay. Oh, yeah, well, thank you. I mean, everyone to hear about Brit's talk on our deep sea chondrichthyans. So Brit is originally from Canada, that has been based in New Zealand for some time now, where she completed her PhD at Victoria University of Wellington in 2017, and she now works as a fisheries scientist at the National Institute of Water and Atmospheric Research. And throughout this period, Brit's become synonymous with all things deep sea chondrichthyans, playing a big role in not only highlighting their existence, but also their concerning conservation status. And within the community, Brit's a member of the Oceania region's IUCN Shark Specialist Group and is chair of the IUCN Deepwater Chondrichthyyan Working Group. She's also president of the Oceania Chondrichthyan Society and was recently awarded the prestigious Eugene Clark Award by the American Elasmobranch Society, recognizing her pioneering and outstanding research on Deepwater Chondrichthyans. And lastly, just in case you thought she might be slacking in her downtime, Brit is also currently the New Zealand women's middleweight Muay Thai champion. So as imposter syndrome slowly kicks in for the rest of us, I'll hand it over to you, Brit.
Thank you, Mick. Thank you for the introduction. I suffer from imposter syndrome every day, so you know we're not alone here. But yeah, thank you for the introduction. I've kind of added a quick slide myself, so I will bypass this. The only thing I will add is that I was once called an itinerant chimera hugger, which I quite like. That kind of stuck. And I didn't realize that was actually a thing until I started going through some of my photos and realized that that is actually what I seem to do. And I had a question before this started, what I do in some of my photos. So these kind of photos you see here, this is me holding a giant chimera. It's actually a small giant chimera. This guy weighed about 12 kilos, but it was also male. We know the females get bigger as well. So yeah, it wasn't too large, but this was taken out on a research vessel. We do fisheries surveys around New Zealand, and every so often I'll go out to sea for about a month at a time, where basically we are counting and measuring a lot of fish, a lot of sharks, and just generally taking an inventory of the deep sea environment. Now, the deep sea environment is fantastic. It's the largest and most complex ecosystem on the planet. It holds the greatest diversity of organisms, and it's also estimated to actually have the greatest biomass of individual organisms on the planet. So it's a very important ecosystem on our planet as well. However, it still remains one of the most understudied environments. Deep sea research is fairly new. However, you know, we're coming up to the 150th anniversary of the HMS Challenger, which was the first kind of survey that actually went around and started understanding our deep sea environment. But we still got a lot to learn about what's going on in the deep ocean. And there is increasing interest in its use. It has been used as a food source, predominantly for fish, for many, many years. We are seeing a lot more interest in using it for energy, for technology. Though that image on the bottom there from Nautilus Minerals, which is actually a bankrupt company now, but those heavy machineries, those are for deep sea mining, and that's kind of one of the hot topics today in the deep sea environment. Deep sea is kind of thought to have these huge deposits of minerals like manganese, nickel, copper, very important very important minerals that are needed for things like electric cars and solar panels. And there's a lot of interest in trying to extract these. In the South Pacific here, we've got the Clarion-Clipperton zone. There's a lot of interest there. It kind of borders some of the Pacific islands like the Cook Islands, Kiribati, Nauru. And we are seeing these nations, Nauru being one of them, that is starting to give licenses out to explore the feasibility of deep sea mining in that region. So it's important that we actually really understand what's going on in our deep sea environment before we kind of, start using it more than we already are. When I talk about the deep sea, kind of depends on where you are in the world, but generally it's considered depth of greater than 200 meters. That's what we kind of consider a deep sea shark, ray, or chimera, your condrithians. These animals here, they get down to generally no more, most kind of, don't get any deeper than 2,000 meters, but there are some that have been reported at depths of 3,000 meters, and the maximum depth from recollection is just over 4,000 meters of a little skate found in the Northeast Atlantic. So it's a huge range of depths that these animals can live at. Now, when we are considering deepwater chondrichthyans, there are species that we know, they dive down to 2,000 meters. Whale sharks do it. Manta rays go down to 700-800 meters. I'm wrapping up a paper that's talking, we're looking at macos that were continually diving to 1,000 meters plus. But in these cases, these animals are not wanted. They're not allowed to hang out in the deep sea all the time, they don't hang out in the deep sea all the time. What we want are animals that are spending the majority of their life cycles at depths of 200 meters plus. So when we're talking deepwater chondrichthyans, we're looking at these kind of guys. They huge diversity of species. They come in all shapes, sizes, colors. Some people find them quite cute. Some people find them horrifying. I think they're fantastic. They've got a huge range of characters that have allowed them to adapt to the deep sea environments. And I'll pick out a few of the more interesting facts as I go along here. So deep sea species, they account for nearly half of our diversity of sharks, rays, and chimeras. To date, there's about 570 or so species, and that continues to increase regularly. Most are very poorly studied. We know very little about a lot of them, and they are very underrepresented in the literature. I was just wrapping up a review with some colleagues, and we looked at chondrichthyan literature within the past decade, and of the work that we found published on sharks and their relatives, less than about 4% of those were on deep water species. So we really need to up the game. We need to get them more represented in our work. We need to stop handing out all the money to white shark research. But why we can't, or why there isn't a lot of work on deep water sharks, there's a variety of reasons. Some species are cryptic, some species are very rare. This pocket shark here, it's known from one individual. So it's kind of hard to really quantify anything about these animals when you don't have large sample sizes, and sometimes it can take years to actually acquire a decent number of animals to better characterize their life history, where they live, those kind of things. Deep water species, they're generally more difficult to access than, say, inshore species. Not only are they found in the deep ocean, but they can be found at locations that can take, weeks to get to. So it can be quite difficult just to get access to them, which also makes deep sea research generally quite expensive. Requires a lot of resources. If you're going to take a research vessel out there, it requires a team of experts that know not only the environment themselves, but how to run the vessel. It requires specialized gear. And yeah, these are all things that cost money. And it'd be fantastic to take submarines out all the time, but who's going to pay for those at the end of the day? So a lot of work on deepwater chondrichthyans ends up coming from opportunistic sampling. As I mentioned, we have fishery surveys here. that go out primarily to assess the status of our commercial species, but we also do look at the environment as a whole. So this is an opportunity of when we can actually kind of study some of the deep water species. We also rely on fisheries themselves a lot of times for samples as well. Historically, deep water sharks have had little value compared to commercial species, for example, so they often receive little priority when it comes to what kind of research we do. There's also been a case too that, you know, deep sea species are generally out of reach of a lot of fisheries, so they generally have lesser threats and could be assumed that, you know, they should have a lesser priority when it comes to actually allocating funding resources to studying sharks. But as I'll show you a little bit later, that's not always the case. But what I'll do is I just wanted to highlight some of the recent advances we've had in the past, you know, 5 years or so. There's been a lot of exciting deep sea shark research, and I hope we can really continue that. Starting off with our diversity. Deepwater sharks, you could describe most of them as black and kind of uglyish, which has been a pain in the butt when it comes to actually monitoring these animals. Historically, a lot of catches were put in an aggregated code called black shark, which makes it really difficult to actually understand, you know, what kind of impact we are potentially having on these species. So recent efforts has really spent a lot of time to try and actually figure out what species we have in our waters. These are the gulper sharks, which are notoriously known for being incredibly difficult to identify to the species level. I'm really thankful in New Zealand, we really only have one, we have 2 species, but only one to really worry about where we study, because they look incredibly similar, and they can also look quite different when you're comparing juveniles to adults, which makes it even more complicated when actually trying to figure out what species you're looking at. Some of the work we're looking at now or hoping to start to do is to really tease out our Denia species, so our shovel-nosed dogfish. We do know we have at least 2 species here. They are one of the primary bycatch species by weight and by numbers in some of the fisheries in New Zealand and outside in the Spriffmo region. But catches is predominantly just assessed as the primary species, which is our shovel-nosed dogfish, Denia calcea. But we do need to understand the cryptic species as well, the less common species, and figure out what we're actually looking at here, because if we aren't monitoring these animals to the species level, there have been cases where cryptic species have started to quietly disappear. New species, the Indo-Pacific is where it's at these days when it comes to describing new species. We've had quite a bit of diversity of chimeras. These are 3, actually two images of the same species, but there were 3 recently described new chimeras south of Madagascar. These were taken from fisheries that were operating in the area. We've also kind of teased out or solved some taxonomy within the Australia region and kind of Indonesia and Papua New Guinea. Again, you're kind of looking at these species that we thought were 2, possibly 3 or more, but looking kind of at the genetics and morphology of these animals, turns out that they are just the same species. So 2 species that were currently known from Australia were combined into 1. And this has implications for management. So now you're looking at one kind of larger species that is also found beyond Australia, which has some implications for international management. But yeah, we're still continuing to sort out these kind of bits and pieces here and there. And there's taxonomists like Dave Ebert, Will White, Simon Wegman that continue to do this work throughout the world. And we're seeing it, like I said, regularly. We've got new and interesting species like this pygmy dwarf shark, sure, I'm not going to get that right, is Planonasis, which came from Sri Lanka, India. a couple years back that was caught in some of the deep water artisanal fisheries there. We've got a bunch of new lantern sharks that are coming out. These guys were all kind of coming from the Pacific region. And again, going back to the review of literature that I've been doing lately with some colleagues, squallus. Squallus are the winners here. In the past, since the last review of our deep water species, we've had 11 new squallus species described throughout the world. I did not include them all here because they tend to all look just about the same. But there are some squallus lovers out there, and they'll be very pleased to hear that we are describing new squallus, new spur dogs around the world. Now, sharks are known to have conservative life histories, and the deep sea is no exception. They tend to have even lower productive life histories, with many of them having very low fecundity, late maturation. They grow very slowly, and they have low rates of population increase. A friend of mine, Cassie Rigby, who's a JCU alumni, she did a fantastic paper a few years ago that kind of synthesized everything we knew or everything we know on life histories of deep water species. And we found that as you increase depth of species, we are finding that generally you're also increasing maturity and longevity, and you're decreasing fecundity and breeding frequency. So as you go deeper, sharks generally have characteristics that make them more susceptible to potentially from fishing interactions. But this is we've got kind of all spectrums here when it comes to what we're looking at with deep water species, right down through the gulper sharks, which are kind of known to be probably the least productive sharks out there. On the left-hand side, you'll see gulper shark that was taken in a fishery of Sri Lanka. That big yellow thing in the middle there, that's a yolk, or sorry, that's an egg yolk. Some gulper sharks, they have one pup per cycle, and when you take into account their age and maturity, their gestation period, which is thought to be 2 to 3 years, and how long these animals are living, it is estimated that these animals can have 12 pups in a lifetime. So that's kind of it's very characteristic of mammals rather than fish themselves. So they are incredibly vulnerable to fishing. On the other spectrum, we've got some species like the Greenland shark, some of the hexanchids as well, which we think could be potentially quite fecund. Problem is that for the Greenland shark, for example, we've never actually caught or seen a gravid female, so we don't actually know what their fecundity is. However, looking at the number of eggs they're able to produce, it is possible that they are producing 300 plus pups per reproductive cycle. So if you're taking into account that these animals could presumably live for hundreds of years, that is potentially a lot of pups that they could produce in their lifespan. Now, when it comes to aging sharks, especially deep water sharks, this is a big knowledge gap. Sharks in general, kind of the traditional method of aging sharks has been looking at growth rings, which you see on the vertebrae. On the left-hand side, you've got an image of a vertebrae, a central vertebrae from a blue shark, and the numbers kind of indicate the growth rings through time. On the right-hand side, you get the same kind of centrum from a deep water shark, one of the plunket shark we have here in New Zealand. And you'll see that there are no indications of any sort of growth rings. So we can't age these species with what we've used to age other sharks. And in turn, especially in the last decade or so, we've tried a lot of different methods. We've looked at a lot of different body parts. We've looked at dorsal fin spines. We've looked at eye lenses, as you might have recalled from the Greenland shark, and their potential to live for 200, 300, 400 years. Eye lenses were used to age those animals. They've also looked at neural arches, tridor ridges, which are found on tooth plates. They've looked at that in chimeras. They've looked at caudal thorns in skates. And most recently, there was a study that looked at telomeres. And all of these examples, they've had varying degrees of success of being able to age animals. But the problem is that we don't have any validation of the study, so we don't know how accurate these counts are. But again, there have been some advances. There are some new technologies. Near-infrared spectrometry is promising. We have been able to do some mark recapture studies. This is from, depending on who you ask, Squallus acanthius, this mighty dogfish, may or may not be a deep sea species. But they, in the Northwest Atlantic, they were recently able to confirm that growth bands that they were seeing on the dorsal fin spine do correspond to age. So you can kind of see there with the arrow there, there's a tiny, tiny little fluorescent mark, which comes from oxytetracycline. It's an antibiotic, actually. It's a chemical that is injected into sharks, and it binds to calcium within hard parts of sharks. And you can use that to kind of validate. Sometimes you can use that to validate age and growth. So we are seeing that we can use it for some of the more, say, shallower deep water species. We are trying it now with some Antarctic skates. Currently working on this species here, one of the amblyarias that's caught in the Ross Sea as bycatch in the toothfish fishery. And we're trying to look at the feasibility of validating growth rings from the caudal thorns. So the caudal thorns are these big thorns that kind of that sit along kind of the back, right down to the right down the tail of the animals themselves, which is kind of indicated by that red box there. And we've injected these animals with OTC. They've been at liberty for at least a year now, and the image on the right is an image of a caudal thorn. So far, we're not having any success here. Well, doesn't look like we're having any success. We are getting fluorescence, but what we're probably looking at there is autofluorescence from the tissue rather than a mark from the OTC. But you can see there are some really great bending patterns within the structure. So we are hoping with another couple of years of sampling that we'll be able to get some more animals, and we'll take a look, and hopefully we'll be able to actually pinpoint some better markers and be able to validate some age with these guys. But in the meantime, our mark recapture study, we are getting good numbers back. It is telling us about the animals we're getting back, is telling us about how slow their growth is. Without saying too much, they are very slow growing animals. They're also not moving very much either. So we are getting an indication. We've got some animals that were at liberty they were originally tagged about 10 years ago, and they've been picked up just about the same spot that they were first captured. So it doesn't look like these animals have very large home ranges either. So there's some great information that's kind of coming out of that project. Now, one of probably the biggest advances that we've had with deep water sharks is bioluminescence, glow in the dark sharks. We have known for some time that there's the Mopteridae family, the lantern sharks, that we know that they glow in the dark. And recently, we've also been able to identify bioluminescence in a new family and some new species. So the image that you're looking at there is a seal shark, Delacius leica. This work was actually done on the last survey which I was out on, and we were actually able to see the bioluminescence right in front of us, which was really cool. And this is really neat too, because the seal shark is, or the kitefin shark, depending on what part of the world you're in, it's now known as the largest vertebrae in the world that can bioluminescence, which is really cool. So there's a lot we don't know about sharks, but we do know when it comes to bioluminescence as well, is that they've also got a very unique way of glowing. A lot of other species that use bioluminescence, they use a symbiotic bacteria that is able to glow. But in this case, deepwater sharks are actually regulating their bioluminescence with hormones. And there's been about 4 hormones so far that have been identified. And they seem to differ across species. So there's a lot more work that needs to be done there. But we are being able to identify it in a lot more species. As you can see here, we've got 6 different species from different families, from different genera, and you can see that each species has a very unique pattern. So they're probably using bioluminescence as some sort of communication. It's also been suspected that bioluminescence can be used kind of as a defense mechanism. The pocket shark, which you saw an image of before, it's called the pocket shark because it's got these little pockets behind its pectoral fins, which they think it kind of expels this bioluminescence to kind of use as a defense, sorry, as a defense mechanism to escape predators, kind of, distracting them with the bioluminescence as it gets away. The other thought too is that they've found patterns of bioluminescence around the spines of some of the deep sea sharks. So they've been calling this the lightsaber. And it's been thought that this kind of advertises to potential predators that, hey, I've got the spine, so you don't bite onto me, kind of thing. So it's used kind of as a deterrent as well. It's also thought that bioluminescence might be used as counter illumination, counter shading, sorry, and could potentially be used for hunting purposes as well. So really interesting stuff coming here. There'll be a lot more soon, I'm sure, as we identify it in new species. So stay tuned. It's a really exciting field of work in deepwater shark research. Another big advancement we've seen in recent years is in movement. Our means of actually looking at these animals, of studying their movement patterns using telemetry and acoustic technologies has improved significantly in recent years. Satellite tags, the major brands are now rated, their tags are rated to 2,000 meters, and most of them come now with this release, this emergency release that kind of gets triggered when tags hit 1,800 meters. So that hopefully prevents tags from being crushed, make sure that tags get back to the surface, so the information is transmitted back via satellite. So there's some super cool ways of actually starting to understand what sharks are doing down there. Now to date, we've only kind of looked at 10 or so species with either acoustic telemetry, or sorry, acoustics, or satellite telemetry. It is, we do have a lot of deep water species, but actually being able to tag these species is a whole other factor. So it'll be quite some time before we're probably able to actually look at a lot more species. But there are more studies coming out. I know there are a few that are currently underway around the world. However, what we've seen so far is that just about all studies have been done in the Northern Hemisphere. So the Southern Hemisphere really needs to pick up its game. We need to get some more tagging work done out in our neck of the woods. Now the acoustic studies that have been done, there's 2 primary ones that I know of. There could be more, but the 2 main ones, one was some fantastic work done by Ross Daly on the southern dogfish. Now this is a conservation dependent species within Australia. Ross set up an acoustic array around southern Australia off Coffin Bay, I believe it was, and he was able to tag 71 animals and got information back from, I think, all but 9 of them. So it's pretty successful tagging right here. And what they found, and what's also been found with this other study here that looked at, it's currently ongoing, and they're looking at kite fin sharks, again, seal sharks, if you're from this side of the world. But with both studies here is that we're finding that these animals actually don't move very much. These gulper sharks off of southern Australia, they were kind of they stayed within the 15-month period of them being tracked. Majority of them stayed within kind of that, it was about 100-kilometer range of the acoustic array. And what they're finding with seal shark around the Azores is that these animals are they're highly residential to the seamounts that they're first picked up on, which is giving us some great information on home ranges for some of these species. Now, acoustic arrays aren't going to work everywhere. It's not something we could probably do in New Zealand, just because New Zealand has such a large EEZ. But places like these have been fantastic. Other means we can start looking at satellite technology. The blunt nose 7 gill, which I see that's spelt wrong. It should be 6 gill, not 60 gills shark. That is another frequently tagged deep water shark species. These are 2 studies here. One was done off of Spain, the other was done off of Hawaii. And we're finding one that's this animal, off of Spain, they really like to stay around kind of a 900-meter depth range, regardless of day or night. Of Hawaii, they had quite different. They were engaging in diel vertical movement patterns, so they were moving up and down the water column. They kind of went over a broad range of temperatures and depths, as well as oxygen levels, but they seem to kind of like hanging out in the low oxygen level, oxygen saturation levels, which is quite interesting. So we are starting to be able to paint a picture of what these animals are actually doing in the deep sea. And with this satellite telemetry as well, we're kind of giving an idea of what kind of or how far these animals can move. Both of these studies, we have one on a gulper shark, one on Greenland shark. Over the course of about a month and a half, some of the animals were moving about 500 kilometers in both studies, which is quite a distance for deep water shark. Now, the problem with tagging deep water sharks is that we don't exactly know how they got from point A to point B. All we have is the location of when they're tagged and the location of where the tag is popped up. But some inferences can be made about how they traveled based on the topography, based on currents. So they kind of get the best estimate of how these animals are actually moving in the deep sea. But it's starting to give us a picture of just where they're going and what they're doing. When it comes to tagging sharks, tagging any shark, really, there are factors you have to consider. It's a stressful event for a shark to be captured and to be handled and then to be released. And even more so with deep water species, because you also have to consider that these animals come from environments that kind of have a lower temperature, they're under extreme pressure, and they have no light. So when they're brought to the surface, this is a very big change to what they're used to, and it can be lethal in some cases. Some species seem to be quite hardy. Again, we're looking at the 6-gill shark here. They have been brought up from 1,100, 1,300 meters and seem to be absolutely fine. There's been other species, particularly smaller species, that have been brought up from a couple 100 meters, and they don't seem to do quite well. So there's a lot we still need to learn about how we can actually tag these animals and bring them up safely, or the alternative is to actually go down there with the sharks and try to tag them from what was done here in a submarine. So Deem Grubbs, there's a video that came out a few years ago, got some funding to actually go down a submarine. And you can see in the bottom right-hand picture there, these kind of pointy, jabby things that stick out. Those were essentially little pressure gun that was used to successfully tag a 6-scale shark from, I believe it was about 600 meters. So that's another way to do it too. Another study way back in the day had transponders that they wrapped up in fish and put them down the bottom of the ocean, and some of those got picked up with sharks. So that could be a potential option as well. It's kind of like, it reminds me of what you do with a dog. You know, if you want to give them a pill, you wrap it in some food and then shove it down their throat. So that's, you know, we could try that some more with sharks as well. So that's, yeah, so we have had some advances here. The other issue when it comes to actually tagging sharks is that, deep water sharks anyway, that once you've brought them up and they seem to be okay and you've got the tags on them, you've also got to get them back down to the bottom of the ocean. And, you know, if you've brought them up from 1,000 meters, they could be disoriented, they could be exhausted. They're a lot more susceptible to being predated on when they're heading back down to their environment. So we need to think about that and how we can get them back safely. There was one study, and I think there's a video that will work here, that trialed a little cage, which essentially they brought the shark up, they tagged it, they put it in the cage, sent the shark back down to the bottom of the ocean in this cage, as you can see here. Once it hit the bottom, this is a little squallus, squallus cubensis, and you'll see these sharks swim off quite happily once it gets back down. So yeah, so we've been quite creative with how we can tag these animals and how we can better understand them. And I'm really looking forward to seeing what we can use in the future to continue understanding these animals. I'm just going to quickly look at the following. Cool. Yeah, I'll just quickly go through some other advances. We've looked at population structure in some of the species. We are starting to understand that even globally distributed or regionally distributed species seem to have population structure. Probably the better examples we have here are individuals in the Northeast Atlantic and Mediterranean. They seem to be separate populations, and that's separated by the Strait of Gibraltar, which actually gets a lot more shallower than some of the areas. And, you know, that has a lot of implications for how we manage these species. So you're looking at 2 stocks rather than 1 stock. Another recent advance is parasites. There's a lot more research going into parasites. I have a friend of mine who loves talking about parasites. If anyone's going to get you into parasites, it'll be her. But deepwater, or sharks in general, they are very evolutionary distinct. They are very ancient species, and that goes for their parasites as well. And that can tell us a lot about the community ecology. So yeah, we're seeing a lot more research going into parasites like the ones on the left here. Those are what you see in holocephalans and the chimeras. And the one on the right there is a very unique, it's a parasitic barnacle that attacks some of the deep water species and feeds off their flesh. Now, sharks, of course, are a very important resource as food and income for communities all over the world. Deepwater species are no exception. Targeted fisheries, we know a lot of targeted fisheries kind of date back to the 1960s. Some go back even earlier. There were bramble shark fisheries in the Mediterranean, Northeast Atlantic, going back to the 1700s. These seem to peak in the late 1800s and shortly decline thereafter with the advancement of steamships. We actually think that the bramble shark is now locally extinct from a lot of regions in the Mediterranean and the Northeast Atlantic. Bramble shark continues to be fished elsewhere. There have been big fisheries for it in the off India, a little bit off Sri Lanka as well. They have been caught off Brazil. And they do seem to be overexploited now. As I mentioned previously, deepwater sharks are really susceptible to fishing because they have really low productive lifestyles, so they can't reproduce fast enough to replace what is being taken out by fisheries. And one of the kind of unique fish targeted fisheries we've seen with a lot of deep sea species is the liver oil fishery. Liver oil fisheries, they have been small scale, they have been large scale as well. They've primarily targeted species like the gulper sharks, and they are looking for squalene. Squalene is a substance that we use in pharmaceuticals. There's been a lot of talks of it using it as an adjutant, as an additive in vaccines, which has become quite topical with COVID vaccine. We've also been seeing it used as biofuels, and it's just generally used as an oil itself. As you can kind of see from the left-hand side there, those are livers being boiled down, and that liver will be extracted and used for kind of local community purposes. Yeah, and unfortunately, these kind of fisheries have decimated, particularly with gulper sharks, they've decimated these populations kind of throughout the world. And what happens is that what we're kind of seeing is that these fisheries, they start up, they last for about 2 or 3 years, and they become really productive, and then they drop off very quickly, and generally collapse and close within about 20 years of the fisheries first operating. Yeah, and you know, when they stop being able to use one species, then they have been moving on to another species. And what we saw in the Northeast Atlantic is that when stocks of some deepwater sharks, the sticky sharks, what they called them, when their numbers started to decline, they actually put in no retention bans. And instead, they started going after and using more chimeras, the ratfish, liver oil, as an alternative to the shark, deepwater sharks. So yeah, there have been a lot of species that have been involved in these fisheries. Unfortunately, most of them, we don't have good information. Monitoring generally either doesn't exist or it comes in after the fishery has happened already. We do have a couple of examples. We can look at things alternative to fishes from catch rates. We can look at things like exports, which was done in the left-hand side there. You see exports of shark liver oil from the Maldives, which go from the 1980s to early 2000s. Again, they peaked really, really quickly, a couple of years within introduction of the fishery, and declined very, very quickly. And we've seen that in a couple other places, including some of the Pacific Islands as well. There was interest in developing these kind of fisheries, particularly in the 1990s, when shark liver oil was really popular. But yeah, in the most run, they weren't economically feasible long term. Probably one of the best examples we do have of documented declines of deepwater sharks took place actually in Australia. Ken Graham's paper was a fantastic comparison of catch rates off Southeast Australia in the late 70s and again in the late 1990s. And for some species, catch rates declined by over 90% within a 20-year period. So some species, they virtually disappeared from those areas. The good thing is that a lot of targeted fishing for deepwater sharks has stopped. In some places, these are protected species. There are no retention bans, which are great. They are species of concern in some of the regional fisheries management organizations, RFMOs. But deepwater sharks continue to be caught as bycatch in a lot of fisheries. As you can see here, so this is primary threat to these animals here. Again, information is quite limited in a lot of fisheries. Sometimes it's not recorded at all, or sometimes when it is recorded, it's recorded under an aggregated catch, again, like deepwater shark. So it becomes very hard to actually monitor how, if we are affecting these animals. And I'll just quickly touch on, this is a complete coincidence when I agreed to do this talk and the release of this paper. But as you might have seen today, which was all over Twitter feed, We have a new paper that's come out. We have reassessed the conservation or the extinction risk status of all sharks, which included 572 deep water species. This did account for 48% of all chondrithians. We have found that 11% of deep water species are threatened, and nearly 20% of all the threatened chondrithians in the world are found in the deep sea. As you might have recalled previously, deepwater sharks generally data deficient. You would have heard years ago that up to half of all deepwater sharks are in the data deficient category as defined by the IUCN. That has been reduced to about 20% now, but it is still quite high compared to coastal and pelagic species, where we're looking at 9%, or in the pelagic case, there's no data deficient species left. So that means that we don't have enough information to assess the status of these animals. And coming from this work too, we are able to kind of see a better picture of what species are in need of research. And as I mentioned, gulper sharks, most species are now at a very high risk of extinction, and that is primarily due to that liver oil trade. There are some skates as well too. There's some big trade for skate wings, which are very popular in some Asian countries, and that has led to the decline of some deep water skates as well. Some other things that we've kind of touched on in this paper includes deep sea richness, so where we find our deep water sharks. Northeast Atlantic is a great spot to find deep water sharks. Japan, Taiwan, conveniently for me, New Zealand, Australia as well is a hot spot for deep water sharks. And what's interesting too is that the Caribbean also seems to have quite a diversity of deep water sharks. And when we look at where sharks are disproportionately threatened, no surprise really there that the Mediterranean Sea has a really high proportion of threatened deep water species, off Brazil, Sri Lanka, India, Indonesia, and Japan, Taiwan as well. Quite a bit of deep sea threatened species there as well. So we have a better idea of, we have a better picture of which species are threatened, where they are threatened, and now kind of looking forward. And as far as, if you want to work on deep water species and what information we need, well, there's still a lot of information gaps. Life history, we still need to figure out how to age these animals, especially the chimeras. We can't find a way to actually age the animals. So we need this information to input into kind of stock assessments and fisheries management tools to better understand just how vulnerable these animals are potentially to fishing interactions. Habitat use, movement patterns, identifying areas that are critical for these species, like egg laying grounds, nursery areas, where they move and when. This will give us a better idea if we need to develop spatial or temporal management. Having this information is very helpful. We can also look at overlap with, say, fishing or deep sea mining interactions, and this will give us a better idea of how at risk these species are to human activities. Post-release survival at vessel mortality, it's virtually nothing. Actually, I've been looking for this for a little while now. We virtually have no information on these studies. No retention bans are fantastic, but as I mentioned, these species are still bycatch in a lot of fisheries. And if we're not allowed to retain these animals, but they are still captured and they come up all dead to the vessel, that kind of defeats the purpose of having a no retention ban. So we need to actually understand, are no retention bans actually useful for deep water species? If these animals are coming up alive and we release them, are there ways that we can release them and so their survival's increased? These are all very interesting questions that we kind of need answers to. We also need to monitor population recoveries in closed areas. So there are closed areas off of Southeast Australia. It has been estimated that it could take up to 60 years for, say, gulper sharks to recover. in areas where they have been hit hard. So we need to actually look at if they are recovering, because there is also interest in opening fisheries again in these closed areas, which will kind of defeat the purpose of actually trying to allow these species to recover. And we also need to better understand supply chains, why these animals are being traded. So the liver oil trade, the skate wings, we don't have a lot of information on, what species are being targeted or used. We don't know where their products are going, how much they're going for. So we need this information to kind of have a better idea of what is actually threatening the animals. But yeah, I think I've gone way over my time. So I'll just say thank you. Please keep in touch. If you're not a member of OCS and you are interested in shark research and you are within the Oceania region, by all means, please join us. We are a very cool group of shark nerds and we'd love to have you. Yeah.
Wow, thank you very much, Brit. That was like a really amazing talk. Thank you for coming.
Sorry, I didn't practice it at all. I had no idea that was going to be so long.
No, that's all good. We'll have time for a couple of questions anyway. What I'm hearing is you need some people to do some genetics work. That's what I'm hearing out of that.
If you're a geneticist, happy to have you.
Yeah, but having said that, I will pass over to Mick and then we might have a couple of questions if that's okay. Yeah.
Yeah. Thanks again, Brit. Yeah, really interesting. Really good to see such a wide range of research methods too being applied. It's really interesting stuff. So I think with questions, if people in the audience, you can turn your camera on and ask Brit. You can type it into the chat and I'll read it out for you. And feel free to use the raise hand function. So I've got a question to kick it off. So I guess similar to like the freshwater things I study is there's this situation where we don't know much about the life history of these animals, yet we can't sit around waiting for that information because they're going to be even sort of in a worse conservation state by the time that's available. So what are some things, I guess, from a conservation action perspective that we need to get going with this group?
I do think that aging is very important, actually getting a grasp on exactly how long it takes for these animals to mature, how long they are living, and validating some of these ages. That does have a huge implication for fisheries management. But again, like actually being able to validate is a very difficult thing to do. If it wasn't difficult, it would have been done already. But as I mentioned, like there have been, and it's trickier with sharks, but something like near infrared spectrometry has been successful with some teleos, and we should be looking at it more. I know Cassie's somewhere in there, and Cassie will be all for this, I think. But Cassie did some fantastic preliminary work on that method, and there should be more done on it. I think it has potential. Yeah, and we do need to kind of ramp up that kind of work. Unfortunately, that kind of work is your basic fisheries research. It's kind of boring. It's not, it's not going to grasp headlines. So it can be quite difficult to fund. I've banged my head against the wall quite a bit trying to get some of this work off the ground. So a lot of it kind of now relies on students to kind of pick up this work. So yeah, and that's great projects for students as well. It's really applicable science, and it's really useful science.
Has there been any, I've sort of been getting into microchemistry recently, and we got a project we're hoping to get funded, sort of looking to try and validate band pairs through chemical signatures, basically, like if we can identify wet seasons or that type of thing. Has there been any microchemistry work on deep sea animals?
There's one example that I know of off the top of my head, which had some interesting results. It was done on a landwind shark in the Northeast Atlantic at Mopterus spinax. And that seemed to have some potential as well. It did show that this really looked at the vertebrae chemistry, and it did show that there was mixing across the wider region, but there did seem to be actually some sort of population structure kind of on a finer scale. So yeah, that is something that could be looked at.
Yeah, very good. Right. Let's see, David's just put your camera on. Do you have a question there?
Yep. Hi all. Hi, Brit. Very nice to see you again, especially talking about our deepwater sharks. And I do have a question. You were talking about mapping the supply chains, and I was wondering how we as scientists can actually do that. There's any methodology that you know to how to study those supply chains?
I think that's more of a social science question, which I'm not a social scientist. But I guess if you were working on kind of small scale fisheries, that is something you could take into consideration, because especially with some of these deep water species, with liver oil fisheries, they can be quite small scale. So kind of, I think that's going to be the tricky thing with some of them, is that they are small scale. There's not international trade. But because these animals are so vulnerable to fishing, that even on a small scale, that these fisheries can have quite an impact on them. So I think that's something that needs to account for, if that can be integrated into research somehow. I'm not the best person to ask about that. I'm not sure if there's anyone here that kind of knows that better, but that is something that we do need to look at. Nice to see you up so late.
Okay. I'm so happy to be here. Thank you, Brit.
Okay, if we haven't got any more questions, I got another quick one. Around the liver oil trade, is there any way, I guess, is there a particular property of the liver oil that makes it more attractive to, I guess, like your vegetable oils or other cheaper, sort of more accessible oils that are available? And if there is there sort of any research into synthetically making it to sort of subsidize the, I guess, natural resource impact?
Oh yeah, there are synthetic versions. There are plant versions, and they, as far as I know, they essentially do the exact same thing. And I think it kind of, part of it would be that now is that some of this shark liver oil fisheries would be a byproduct of other fisheries for maybe potentially more sustainable sharks, which does further complicate things of actually figuring out, you know, what sharks should we kind of be using and versus what sharks shouldn't we be using. But yeah, no, definitely there are alternatives. But I think for especially kind of local use, it might just be because it's accessible. They use it there. There might still be that kind of appeal that you know, you gotta have the shark one, you can't have the synthetic one. But yeah, there's definitely alternatives out there.
And so it is largely very much local consumption then, as opposed to international trade where CITES and could be options.
Yeah, I think so. I think today, especially back in, say, the 90s, there was a lot of international trade, but that did dry up. There wasn't oversupplied to the market in the, I think, about the mid 1990s. And that seemed to kind of, I don't want to say crash the market, the international market, but it definitely affected the international market.
Yeah. Okay, well, we've got about 1 minute left. If anyone has a last opportunity to ask a question.
Brit, it's Maddie Green.
Hi, Maddie.
Hi, great talk. Thank you. Loved it. I was wondering, and I love asking people this question, but if I gave you like $1 billion, what would you do? Like, you know, what kind of research, you know, would you work on the most? Would you, like Dean Grubbs, get in one of those submarines? Like, what would you do, and how would you help?
Me personally, I'd love to do some movement work in New Zealand. I would like to do a telemetry study. I would like to combine it with genetics, as I've been told from geneticists that we need to look at both kind of movement from a satellite tagging perspective, but also look at the genetics. So that's what I would do. That's what I've been trying to do. I've been trying to get funding for that for a little while now. Yeah, I think it's quite important to we need to understand what these animals are doing, because there is a lot of interest in assessing spatial risk and overlap of fishing distribution and distribution of the animals. But I don't think we have sufficient information to actually properly assess where these animals are in the water column, where they are spatially, and what they're doing throughout the year. So I would like to do that.
That sounds fab. Maybe we can take some funding for some more from some charismatic species and maybe use it on some lesser known.
Maybe get on Shark Week, Brit.
I'll sell my soul one day.
All right. Well, we might let Brit off the hook there and wrap it up. So yeah, if you can all join me in giving her a big virtual round of applause. Yeah, thank you all for attending.
Thank you. I'll just stop the recording now.